So internal components. So here the internal structure. So it is also similar to the monocot stem. So some differences, some variations are seen in the dicot stem and monocot stem. So here the example, mainly the typical example for this monocot stem is the mage stem. So it is taken as an example. Okay. So here the TS of this monocot stem, which shows epidermis, cortex, and stele. That is here the monocot stem, which also have the dermal, that is epidermis. Dermal tissue system means epidermis, and hypodermal tissues means cortex. Hypodermis and ground tissue system is, that is the cortex, outer cortex, inner cortex, and endodermis. Next one is the vascular tissue, that is stele region. So main difference from dicot stem is the ground tissue, not differentiated into cortex and endodermis. So here, this ground tissue, which is not differentiated into cortex and endodermis. Okay, so it is the main difference. So dermal tissue system, that is, it is composed of epidermis. So it is the outermost layer and single layer, and which is made up of parenchymatous cells. And these cells are compactly packed, and thick layer of cuticle is present on the epidermis. So cell wall is silicified, that is silica depositions are seen on the cell wall. So here the epidermis is persist as long as the stem, that is no secondary growth. Here in generally in monocot stem, that is secondary growth is absent. So epidermis is persist throughout the life, whereas in dicots, herbs, so variation is seen in the herbs and trees. So generally in the trees, that is epidermis is replaced by the outer layer, but here epidermis is persist throughout the life. So hairs, here the trichomes are usually absent. So the main difference is here the hairs are absent. Okay, so next one is here few stomata are present on the epidermis, and here the main functions of these epidermis is limiting layer, that is the outer boundary, and which provides protection. The main function of epidermis is protection. And silica deposition, which provide the mechanical support. So this is also a special character, that is silica deposition. So whereas in dicot stem, that is the cuticle is present, but silica depositions are not there. But here the silica depositions, it is also a special character in monocot. So cuticle prevents the transpiration, and stomata allows gases exchange. Next one is the epidermis, which is followed by the hypodermis. So this one is the epidermis, single layered. So it is followed by the hypodermis. Here it is mainly made up of sclerenchymatous cells. Okay, so here occupy immediately below the epidermis, and here the cells are polygonal and compactly arranged. So it is multi-layered and sclerenchymatous. So generally in dicot stem, that is collenchyma, but here it is sclerenchymatous. So alternate patches of chlorenchyma may be present, and below the chlorenchyma, hypodermis is continuous. And here the peripheral vascular bundles, which are embedded in the hypodermis. The end function is which provides support and protection. So the main function of this hypodermal tissue system is which provides support and protection. So next one is the ground tissue, so which is composed of cortex only. So ground tissue is not differentiated. So it is not differentiated into outer cortex or inner cortex. So here cortex, that is parenchymatous, that is it is made up of parenchymatous cells, and here the cells are large and circular, and cells loosely packed with intercellular spaces. So in aquatic monocots, that is potamogeton, that is parenchyma is present. So in some plants, central portion is hollow, that is in Oryza and Triticum. And here the main function of this ground tissue is which helps in the storage, and here only vascular bundles are present in the ground tissue system. So next one is a vascular bundle. So vascular tissue system, so it is composed of vascular bundles. So here the vascular bundles are numerous vascular bundles, whereas in dicot stem only 6 to 8 vascular bundles are present. But here many numerous vascular bundles are present, and they are arranged in the scattered in the cortex. So many vascular bundles, they are scattered in the cortex. So these vascular bundles are widely separated from each other, and they show size differentiation also. Some are larger in size and some are smaller in size. That is, the vascular bundles in the outer region are smaller, and the vascular bundles in the inner region are the larger. So here the variation in the size also seen, and many vascular bundles which are scattered in the cortex. So vascular bundles are conjoined, collateral and closed. So another difference, that is the closed vascular bundles are seen in monocot. That is closed means here the cambium is absent. So cambium is absent, and here the vascular bundles composed of xylem and phloem, and xylem is endark. Endark means that is protoxylem towards inner side and metaxylem towards outside. And xylem elements are arranged in the shape of V. That is metaxylem elements which forms the arms of the V. So central protoxylem on either side, here these are the metaxylem. So protoxylem elements occupy at the junction of the V. So here protoxylem on either side, here these are the metaxylem. So metaxylem composed of two large vessels, and these vessels are with pitted thickenings, and metaxylem tracheids which are present between the metaxylem vessels. So protoxylem mainly composed of a few vessels. These vessels shows annular or spiral thickenings. So protoxylem elements fuse to form the lysigenous cavity. So they are disintegrated or dissociated to form the lysigenous cavity. So this cavity it is called as a protoxylem lacuna or protoxylem cavity. So here what happens? So these protoxylem, after they are dissociated to form the cavity, it is called as a protoxylem cavity. So this protoxylem cavity which is absent in asparagus. So it is also a special character, that is so special extra character, that is in asparagus, that is the protoxylem cavity is absent. So phloem which is situated outer to the xylem, that is between the V arms of the V, and phloem composed of sieve tubes and companion cells, and phloem parenchyma is absent. So here the vascular bundle in most cases are surrounded by the bundle sheath. So this is also a special character in monocot stem. So bundle sheath sclerenchymatous, that is the sheath which is made up of sclerenchyma, and the bundle sheath is absent in asparagus. So this is about the monocot stem. So the mainly here the epidermis, here the trichomes are absent, and next one is the hypodermis, and here the cortex is not differentiated into outer or inner cortex. In the cortex region only these vascular bundles are scattered. There is no endodermis. Okay, endodermis are pericycle, so these are scattered in the cortex region only. So each vascular bundle which is covered with which is surrounded by a bundle sheath. So these bundle sheath which is made up of sclerenchymatous cells. So this presence of bundle sheath, it is also a special character which is seen in the monocot. So another one is the here the secondary growth is absent in monocot stem. So differences between these dicot and monocot.